Now we are going to start upon the second chapter of immunology, that is several components. In several components, we are going to study first our innate and adaptive immunity. Before that, I have to talk to you about the barriers for infection. If any infection attacks on the body, for example, any bacteria, so first they have to cross the skin and mucous membranes. Second, after defeating this line, they have to go with innate immunity. If they cross this, then they have to fight with the ultimate immune system, that is adaptive immunity. Adaptive immunity. So in this one is sequence. So what is innate immunity, what are adaptive immunity? So the innate immunity having some components, same adaptive immunity also having components. First here going with innate immunity. Innate immunity having neutrophils, macrophages, monocytes, dendritic cells, natural killer cells, complement system, physical barriers and secreted enzymes. On the other side, adaptive immunity having T cell, B cell circulating antibodies. The mechanism in that are germline encoded. Germline encoded refer to genetic information present in germ cells, like sperm and egg cell of an organism which is inherited by offspring, and they have the constant. Okay, but in adaptive immunity there is a variation through VDJ recombination during the lymphocyte development. It's a crucial process in the adaptive immune system where developing lymphocytes like B and T cells randomly assemble different gene segments. That V is a variable, T for diversity, and J for joining to create unique antigen receptor genes. V means variable, T for diversity, and J for joining. By these components, they make a unique and multiple antibodies for different and numerous microorganisms. Now, response to pathogen. In innate immunity is a non-specific response, but in adaptive it is a highly specific and refined over time. It is a little bit fast. It is respond within minutes to hours, but it is a little bit slow, and it develops over long periods memory response. And it is maybe in days and weeks. And it is having no memory and it having memory. And when there is a memory, the response is more faster and more robust. In innate immunity, there is no memory of the response. For example, when microorganism come and innate immunity kill it, but it have no memory and it did not remember the strategy how they overcome that microorganism. When in adaptive, they strategize all the thing, make a strategy for fighting with this. So next time if these organisms come again, then again they will fight with same strategy. Okay, that is like memory. Now we are moving to secreted proteins. The secreted proteins are lysozymes, complement, C-reactive proteins, defenses, and cytokines. And here we have an immunoglobulin with the cytokines. These are key features in the pathogen recognition. How they will recognize the pathogens. In innate immunity, they are having tool-like receptors, and they are also having pathogen receptors that recognize the pathogen and damage-associated molecular patterns that get PAMs and DAMs. By this, there is activation of NF-κB, and by this, they will release of pro-inflammatory cytokines. And we have an example of lipopolysaccharide. In lipopolysaccharide, gram-negative bacteria. Gram-negative bacteria, we have a lipopolysaccharide that is endotoxin. It binds with CD14 macrophages, and also it will trigger first they will go with toll-like receptors 4, and then it will be trigger and initiate interleukin 1, 6, 8 and tumor necrotic factor. Understand? And same like that, we're having more like flagellin and also peptidoglycan. Glycan. It is a presence of cell wall of bacteria. They will attach with NOD receptors, and NOD stand for nucleotide binding oligomerization domain, and they will introduce themselves to the toll-like receptor and NOD receptors. And we also having mannose. This is present in bacteria and yeast, and they will interact and release by liver, that is called MBL, mannose binding lectin. And this mannose binding lectin then activate the lectin pathway of complement activation. And also we are having double-stranded RNA. In human being, the double-stranded RNA is not present, so our immune system easily detect that and attack on this double-stranded RNA of viruses or bacteria and microorganisms. Also, they having non-methylated DNAs. So also in a normal human being, there is methylated DNAs. So our immune system also recognizes and attack that thing. And after these all the things, you guys know that for the gram-negative, we have lipopolysaccharide. Same for the gram-positive, we have lipotacharine acid. Lipotacharide acid. It is for gram-positive. For gram-positive, we having lipotacharinic acid, and for gram-negative, we have lipopolysaccharide. LPS. Understand? This is all about the innate immunity, how innate immunity is going to recognize the pathogens and recognize the microorganisms by bouncing dams, and there are different organisms. But the thing is then, there's a LPS, there's a peptidoglycan, mannose, anything is there, but they will give us same response to every stimulus, every endotoxin, every component of microorganism, because they are non-specific. But in an adaptive immunity, they are more specific. They are having the memories and they are activated B and T cell that subsequent exposure to previously encountered antigen. They have a stronger, quicker, and immune response. And adaptive immune response decreases with age, and it is called aminosis, the aging of immune system. How they will do? First, innate immunity, integrate the macrophages, and macrophages make the phagocytosis of that microorganism that make species of that bacteria, and then they will go to antigen presenting cells. They will go to T cell, and then from T cell they will present antigen act on the microorganism, like that. We will read it in detail in further topics. Now we are having our next topic, that is immune privilege. Immune privileges mean some of our organs having not as much active immune system due to some reasons, and our immune system not exposed to that part of bodies. There were some organs like eyeball, the brain, placenta, and testes. Some reason is that there are some blood barriers due to that, and also they having not exposed to immune system as much. So these cells for chemical physical mechanism limit immune response to the foreign antigen to avoid damage. There will occur from inflammatory sequence due to weight, due to immune privilege to that organs, eye, brain, placenta, or testes. They are having less risk of allograft rejection. They have less risk of allograft rejection as compared to others, because these organs have less exposure to immune system.